Okay, good morning, everyone, and welcome to our first distinguished seminar this semester. And today we have Giulia Preti talking about network harmonics of the brain, a graph signal processing framework to investigate structure function coupling. And Giulia is a senior scientist and lecturer at the NeuroX Institute, EPFL, Switzerland, and is also affiliated with the Department of Radiology and Medical Informatics at the University of Geneva, Switzerland. She received her master's degree in biomedical engineering and her PhD degree in bioengineering from Polytechnico di Milano, Italy. During her PhD, she was a visiting student at MIT and also Harvard Medical School. She joined the Medical Image Processing Laboratory in EPFL and the CIBM Center for Biomedical Imaging in Switzerland as a postdoc in 2013 and became lecturer at the University of Geneva in 2018. So let's welcome Giulia.
Thank you. Thanks a lot for the introduction. And thanks for the invitation and organization. I'm super happy here today. So my talk is pretty didactical, so feel free to interrupt me. I was not sure about your background and crowd, even online. So we'll go through the concepts, and I mean, if you don't understand something or if it's unclear, you can just interrupt me. So today we'll see together a method that we use to investigate brain function and structure with network harmonics, which is based on graph signal processing. So the outline of the talk will be the following. So first, we are going to see what is our goal from the neuroscientific point of view. Okay, so bridging structure and function, why this is challenging, and how we're going to approach that. Then we're going to dive in the methods. So first, processing framework for structure function coupling, the findings. So we have been applied this framework for the past 5 years. So just what are the results that we obtained, and then the clinical applications that we have been exploring in the past few years. So we start from the goal. So bridging structure and function. What does it? So you know probably already that we have a technique, pretty powerful, magnetic resonance imaging, non-invasive, with which you can actually, using different contrast, using different settings of the physician, in order to get different information about the brain. In particular, the type of information that we are going to explore today are two. One is the structure, and here by structure, again, you can have different type of images, you can look into different type of structures. Here we're looking at the structural white matter bundles that connect different regions in the brain. And we can access that information with a technique called diffusion weighted imaging or diffusion MRI, and looking and with a technique called tractography that allows you to virtually reconstruct what are the white matter bundles in the brain. So this is, just to keep in mind, an indirect measure of connectivity because we don't have the spatial resolution in order to really look at the neurons, but we can infer what is the main directionality of the tracts in one specific location. And so we can reconstruct what is a virtual track, basically. And we can see then how brain regions are physically connected. I think I see the whole screen, but here something is missing. I don't know if you want to, probably it's not that, but yeah, if you want to fix that. The second type of information is functional information. So we can access that, again, in many ways. One opportunity to do that with MRI is based on functional MRI that allows you actually to look at the different activations in the brain. So here we're not talking about structure, we're talking about how the brain works, how it activates, and you can retrieve actually brain patterns of activity, like here, red and blue, so like positive and negative activation, or you can look at regions that co-activate and co-deactivate. And so when we talk about functional connectivity. As for the first technique, again, it's an indirect measure of neural activation because we cannot really look at the activations of single neurons with this technique. But what we look at, the contrast, is the blood oxygenation level dependent contrast and look at mechanisms of oxygenation of the blood. So thanks to neurovascular coupling, where you have an activation in the brain, you're also hypothesized to have this augmented cerebral blood flow with oxygenated blood. And so we have an increase in this pulse signal, an increase of oxygen. This is what we measure. So as said, we want to combine these two types of information. It's challenging because even if it's the same technique, they come from different acquisitions. They also reflect different tissues in the brain. Here you are reconstructing white matter. The activations are in gray matter. We would like to do it in a non-trivial way. So just how do we do that? Just a little bit more details. When we talk about the structure, in order to build a structural graph, we need a parcellation. A parcellation is just a subdivision of the cortex into different regions. Many parcellations exist. So the first thing to do is choosing a subdivision, choosing a parcellation, like this one, for example. And then by merging the parcellation with the tractography, so with the reconstruction of all the fiber bundles of the brain, all the boxes, we can actually count, we can look at, quantify the connectivity, in this case structural, between different parcels. So pairwise, you can consider basically the number of threads. For example, there are many metrics that one can consider, but if we talk about the very simple one, number of threads that connect different parcels of the brain. So you can construct a graph, as you see there, where brain regions of your parcellation are nodes. Okay, here they are located exactly in the coordinates of the brain. And edges are represented by the amount of connectivity between different brain regions. Okay, so this is called structural brain graph or structural connectome. You can also visualize it as an activity matrix, but this is what we are going to use as a graph. So the structure. Then, as said, we look into the function. So from fMRI bolt, we get something like that. So this is different cuts, actual cuts of the brain. And we look at the patterns in time. So this is accelerated 6 times, but still you can see that you have a lot going on. And this is a resting state acquisition. So the person is lying in the scanner without doing any particular task, just resting. So you see that we have this dynamic activity, and at every time point that we record, so every volume that we acquire in the MRI, we will have one snapshot, so one of these functional activity patterns. So what do? We use the same parcellation that we considered before, and we can merge with the functional bold volumes by averaging the time courses within each parcel, within each region. Okay, so we obtain n time courses like that, if you have n regions in the brain. And if you consider them, so this is an example of 2, then you're going to look at how each region in your parcellation activate, basically. So this is the bold signal of each region. What you can do is then constructing another brain graph that is, in this case, a functional brain graph. It's a functional connectivity graph. It has the same nodes, okay, so same space of this one, but here edges encode the functional information, okay? So basically, how you just look at the correlation between all these time courses pairwise. So correlation between 2 nodes would lead you to the edge in this brain graph. We are not necessarily doing it for the graph signal processing framework of today, but just to give you the full picture. So normally people reconstruct the structural brain graph, the functional brain graph, and what was done in the beginning, let's say, not mostly now. What can be done is just simply correlating these 2, right? So you have 2 connectomes, structure, function. You can look if they correlate. But you can imagine that the structure just gives you the set of roads of your brain, and then the function is how these roads are used. So obviously function stems from structure, but there is not a one-to-one correlation. There is also not a relationship that is so trivial to be described by one coefficient of correlation. So previous studies showed, for example, a positive correlation, obviously of 0.7, 0.8 between these two graphs, but they don't tell us much about how function relies on structure, right? So there are different ways, obviously, to go beyond correlation and look at the relationship between these two information. Different ways that have been explored in literature. Some assume a model, like big computational models, like the virtual brain, for example. Not, but what we are going to explore today is how to combine these two pieces of information with the framework of graph signal processing. So let me dive now into the details methods. So graph signal processing is basically the translation, so a field that is quite recent and translates operations that you normally have in conventional signal processing, like transforms, Fourier transform, filtering of signals, but it applies to signals that are defined on graphs. Okay. So for the purpose of the brain, we said we have a graph, and from now on, as a graph, we will consider the structural graph. So we forget about the functional graph, okay? We have our connectivity that is given by the structural connectivity information. And then for the GSP framework, you need a signal. What is a graph signal? It's just a vector with n values, so one value per region. So a vector that associates one different value at each region of the brain, at each node of the graph. So it's a signal defined on the same graph, but it doesn't have edges, okay? So again, the graph is coming from the structural connectome with the parcellation that we saw before, and the graph signal, as a graph signal, we're going to use our functional time courses. So for each node, we associate a functional time course that we record from both. What is the catch here? So here is just another representation. You see the time, this is your starting data. Okay? So you have your structural connectome, you have the functional time courses, but one very important key thing to understand is that here our graph signal is not a time course. Okay? So as we said, it's a vector that associates one value per node. So it's like considering one time point. So if we consider one time point here, this is our graph signal that we project on the structural connector. So it's just the brain pattern at time point t. So it has dimensionality of n number of regions. And a piece of information that we have is that it evolves in time. It's dynamic, but we will consider it as having multiple signals on the graph. Another example for you to understand, if it's the first time you hear this, let's say that you have a map of a city, like the bus stops of the city. This could be your structural map, right? I mean, how the different locations or bus stops are connected by different lines of bus. And a graph signal could be, I don't know, anything that you can detect at each node. For example, the temperature at each bus stop, or if there is a vending machine or not at each bus stop. So it's really like one value, one scalar value per node. So we will have a bunch of graph signals to analyze, but we have around 500, 1,000 for a typical resting state acquisition. So again, this is our structural connectome, the graph, just another representation that shows you basically the nodes with a certain parcellation, and our graph signal, an example of a real fMRI frame at time point t. So you see for each node, basically it associates a value of activation. Okay. What do we want to do? We want to investigate how the function is using the underlying structure. And to answer this question that is a bit an outstanding question you know that function you saw from the video before, function moves in time. And so how does the correlation change since we have this dynamic function that relies on a pretty static structural connectome, because the structural connectome doesn't change with the same time scale within the 10 minutes, 15 minutes of acquisition. So how do we explain the relationship between these two? So even the relationship should be dynamic, right? Because we have this function that evolves on this static structure. And we will see how this framework allows you to actually do it in elegant. Okay, so we need a little recap on two main concepts before going ahead into the real steps of the method. I'm sure many people are already familiar with that, but it's good to be all on the same page and to do a little bit. So the first one is the concept of smoothness on the graph, okay, of our signal. So we have, if we start from the definition of total variation of a signal, we have a one-dimensional signal here. What does it quantify? It quantifies the changes of the signal between consecutive points, okay, squared. So if a signal is smoother, so it means that it has basically smoother variations, it has less changes between consecutive points. So this value is low, right? This is pretty standard, pretty easy to understand. Now let's project that into a graph signal. So a signal that is defined on the graph. We have another definition of total variation of a signal on a graph, okay, that is given by this sum squares of some differences between, so this is x, is the signal values, okay, and aij is your adjacency values for the graph, underlying graph, so it tells you how basically nodes are connected. And you can see that this equation here of total variation, what it quantifies is how basically the signal resembles the underlying graph, because it will be low, so a smoother signal on the graph, if two nodes with similar values are connected. So let's put it in the other way around. The two nodes that are connected in the graph, they also have similar values in the graph signal that you are considering, okay? So here, X, again, it's a graph signal. A encodes the values of your structural compute. So yeah, basically a signal is smooth on the graph if the values at two very connected nodes are similar, okay? And the opposite. It's not smooth if instead basically connected nodes, so neighbors, but in the topological sense of the graph here, they have similar values. Not similar values, sorry, the opposite. Then Laplacian embedding. So what is, oh yeah, so sorry. So if the TB is low, so total variation is low, we said you have a smooth signal. We can also make this parallel now with signal processing with the concept of frequency. But now we are in space, you remember? Our signal is not in time, it's in space, so in the space of the graph. So we can also define this TV value as a metric of spatial frequency, because a signal that is smoother on the graph will have a lower spatial frequency, exactly like temporal frequency, like smoother variation. We normally say they have lower frequencies, right? And instead a signal that changes a lot over connected nodes, so over the edges, then we will say it has a high spatial frequency. So this will be important for later. If you have any questions, just interrupt. And you're seeing online, right? Okay, so Laplacian embedding. So now what do we want to do? We look for a space for coordinates, let's say, lower dimensional space, in which the signals that are defined on the graph vary as little as possible across the edge. So we look for a space where our signals are really smooth on the underlying graph, okay? Let's start from this hypothesis. So in other words, if we have the same equation here of total variation, just replacing the signal with u, okay? So differences between signals, adjacency values, we want this to be low, so we want to minimize this quantity. For this, to find the signals u that are smoother on the graph, okay? When we minimize this total variation, basically, what we find is that the vectors u that are the solution and the non-trivial solutions of this optimization problem correspond to the eigenvectors of the Laplacian, defined as a degree matrix minus the adjacency matrix. So if you have the graph Laplacian here, P minus A, for how it is constructed, it encodes, it represents a sort of derivative of your signal in the graph space. So if you project a signal into the Laplacian space, basically you are going to maximize smoothness on the graph, basically. So the Laplacian encodes this variation, and the key factor here that matches these two concepts is that this equation of total variation of signal on the graph coincides with the eigenvalues of the Laplacian eigenposition. So if you actually minimize that, you find that solutions U that satisfy this equation are the eigenvector of the Laplacian. And if you use the quadratic form as here, so with the square differences, then this will coincide with the eigenvalues. So what we are saying, in other words, is that if we consider the Laplacian of the graph that we have, so of our structural connectome, and if we eigen decompose it and consider the use that the eigenvectors that minimize this equation, so the one with lower lambdas, we are projecting our signal in a space that maximizes smoothness on the graph. Just to see it as an example, here is the brain. Okay? We just encode with two different colors, left and right, okay, hemispheres. What we know is that within hemispheres, nodes are more connected. I mean, this is just based on a structural connectivity of the brain. If you visualize this, which is now in the nodal space, in the Laplacian embedding, so just a projection of these nodes into the space spanned by the first two non-trivial eigenvectors, so the second and third, you see that nodes are clustered together based basically on their smoothness on the graph. So nodes that are very connected here, they end up very close by in this space. So we found a space that actually achieves that. So Laplacian eigenvectors U, in other words, span a space where connected nodes are close. So this is just a fact that we're going to use in order to decompose our structural connectivities. Okay, so now you're ready for the pipeline. So as we said, we consider the graph as a structural connectome. We can visualize it as a matrix n by n. It's a symmetric weighted matrix because we don't encode directionality with diffusion, am I right? And also we want to quantify the connectivity. So it can be, for example, the number of tracks normalized, but normally by some constant, for example, the areas of the regions, but you have also many other options, many metrics that you can consider from diffusion. Then, as we said, we compute the Laplacian d minus a, we eigendecompose it, and we look at the eigenvectors. The eigenvectors should be signals that are really smooth on the graph, the ones that minimize this. So here everything is structural, okay? I didn't take my function yet. So the first step is all structural. So let's look at how these eigenvectors look like in a real case in the brain, okay? They're also called structural harmonics, we'll see later, right? And you have here just an example of the first two and then the last. So you will get n harmonics, n eigenvectors. As we said, if we order, they are ordered here by increasing lambda. The first ones will have very low lambda, which means, again, there is a reminder here, very low total variation on the graph. So these low frequency eigenvectors will contain a lot of information about your underlying structural connector. Okay? In other words, they are going to be more coupled to the graph. In fact, you can see that, okay, apart from the first one, which is constant, that corresponds to the zero lambda, okay, that you always have due to the construction of the Laplacian, but that we kind of ignore because it's not really interesting as a solution. If we look at other low frequencies eigenvectors, you see that particular this one is exactly like the example that we showed before. So it encodes the main directions of structural connectivity. Here it separates left to right. Then you will have one that differentiates anterior to posterior and so on. So each of these patterns, each of these structural harmonics, it's associated, to an eigenvalue, and we said the eigenvalues or total variation metrics, they encode information about the spatial frequency. So we can say that each of them basically relates to a certain spatial frequency going from smoother, so lower frequency, to high spatial frequencies. In practice, the one that you have at the other end of the spectrum, so the very high frequency eigenvectors will be more focused, will contain less information about the structure. You are more probable to have this red, blue in terms of signals, even in nodes that are connected, because it means that this is high, okay? Even if nodes are connected, the signal values are going to be different, okay? So in other words, we can say it's signals that are more liberal on the nodes. So while decomposing the Laplacian, so when we look at the eigenvectors, we find basically a set of structural bases for which we know the first one, the low frequency one, they encode a lot of information about structural connectivity, and at the other end of the spectrum, the high frequency one, they are more liberal. They always come from here, but they are more liberal, okay? So we are going to use now these bases in order to project our functional signal into the space of the Laplacian eigenvectors. Okay, so here we take a function, again, functional signal, fMRI frame at one time. Okay, we don't have the temporal dimension yet. So we take this signal here and we project it into the space of the Laplacian eigenvector. So in other words, we do the Laplacian embedding that we were talking about before. We write it then as a linear combination of structural harmonics by computing these spectral coefficients. So these coefficients S hat that are basically, that represent the weight of each structural harmonic present in your signal. So in other words, what are you doing? You are basically considering your signal and quantifying how much of structural patterns in your structural basis is present in your signal at time point t, okay? Because you are estimating or you are computing these coefficients here. Also to say it in other words, you basically project your signal into the graph spectral space that is spanned by the Laplacian eigenvectors. This is the equivalent of doing a Fourier transform for conventional signal processing, and it's called graph Fourier transform. So it's over there. So you write your signal S as a linear combination of columns of U's, or this is actually the inverse. If you start from your signal S, you estimate, you compute this S hat that are your Fourier coefficients, okay? Spectral coefficients of your decomposition. You can see it also with an analogy, because normally what you do with a signal in time, okay, with a time course, you project it into Fourier in order to basically see what is the contribution of each temporal frequency, right? So you write it as a linear combination of harmonics with different temporal frequencies. Here is exactly the same, but you're writing your signal in space as a combination of different spatial frequencies. Another thing, basically here we can answer the question that you were asking before, because you can imagine that when I use a different time point, then I will have the same structural basis, okay, so the same set of structures, but I will get different coefficients. So you see how this framework allows you to have a static structure. So this, you define it in the beginning and you are never changing that. But at each time point, these structural modes will combine differently in order to give you a different functional pattern, basically. Okay, so what we said is we take a graph signal X in the original domain of the graph. We take it to the spectral domain of the graph. So we project it into the eigenvectors columns of U of the Laplacian. What can we do now? So normally in signal processing, when you do that, you want to change your signal, you want to perform some operations on your signal. One that you can do is filter it, right? And exactly as for conventional signal processing, we can use spectral windows for filtering our original signal and reconstructing basically a filtered signal into the original domain. So we can actually choose H as a matrix that basically selects only few eigenvectors of the Laplacian, okay, and reconstruct a signal, so an fMRI frame at time point t, that only includes these vectors, these eigenvectors, instead of all the eigenvectors. I didn't specify, but here, if you don't filter, obviously, I mean, everything is linear. if you go, you can go back to this from the spectral domain to the original domain and reconstruct your signal if you use your full reconstruction, so your full matrix U. But if you cut U, for example, with a low pass or high pass, you only consider the first or last eigenvectors, then you're going to basically reconstruct a filtered version of X that only includes a few eigenvectors. Why do we want to do that? And why this is the third step of the pipeline, actually. So we have our signal decomposition, same as we saw before. Now you see I select a cutoff, okay, certain cutoff in the spectral domain, and I cut my signal into two portions, okay, so with an ideal high pass and ideal low pass. And I reconstruct then a portion of the original signal that comes only from the low spatial frequency, and a portion that comes from the high frequencies, basically. From what we said before, the low frequency eigenvectors are the ones that encode more information about the structural graph. So what do I know about this portion? I can consider that this portion of the fMRI signal is more coupled to the structure, is the portion of signal that comes more directly from the underlying structure, while this is the one that can be explained a bit less with the direct, let's say, directed paths of my structural connectome, and it's more liberal. It's also another term that is used in literature. Obviously, present in the signal and also comes from harmonics of the structure, but that are less smooth on the graph. Okay. So now, for my original fMRI data, for every time point, I obtain two patterns. Okay, so I doubled it. One portion that is coupled to the structure and one portion that is decoupled. I can do it for each time point. Okay. So I will have basically two sets of time courses that I obtain, one that is coupled, one that is decoupled. And then the first thing that we thought of doing was to quantify if some regions of the brain were more consistently coupled or decoupled from the structure. So during your resting state of position, okay, what is the energy across time of these two data sets, basically? And so we did that by computing the norms, two norms across time of the coupled and decoupled portion across time again. And so the ratio between the two, so the high frequencies energy divided by the low frequency energy gives you this index that we call structural decoupling index that quantifies for each node in the brain if the energy of coupling and decoupling was different. And in particular, if it's more often coupled or more often decoupled across time. So you see it's a ratio. So if this value is 1, then it means that the region is sometimes coupled, sometimes more decoupled, but on average, it doesn't have a strong behavior towards one of the two. Otherwise, if it's higher than 1, it's more decoupled. If it's between 0 and 1, then it would be more coupled. Okay. Okay. So. We are here at the functional activity filtering. Just as I said, here is just a representation of each volume in time. What we can do, so this structural decoupling index is just a nodal value, so it's value per node. So it's a nodal measure. Okay. What we can do is also trying to reconstruct an edge-based measure, like the functional connectivity, but a version that is coupled and a version that is decoupled. So later what we did was, okay, trying to understand which part of this functional connectivity comes from either the one or the other. And you can simply do that by computing Pearson pairwise correlations, but based on these coupling or decoupling factors. So you will obtain two connectomes, basically, one that is more coupled, one that is more coupled. You can see by eye as well that coupled functional connectivity resembles a lot functional connectivity. Here is just like selecting the function that comes directly from the structure. And I mean, we saw how the low frequency harmonics were, right? They were full brain, encoding this very well-known resting state, large-scale networks. So it's not a surprise that actually the coupled functional connectome also resembles more what normally dominates functional connectivity. But we have a portion of signal that encodes some other correlations, and the objective aim was also understanding if this is just noise or if it makes sense, right? If there is a meaning in this decoupled portion. It certainly looks more noisy, right? It's like smaller variations, patterns that we don't really understand. So we will see that we also investigated that. Okay, so now you have the full picture. So just to recap, you have your structural connectome decomposition into a set of structural harmonics, each with a special frequency. This is your basis function of the graph Fourier transform. So you take your signal defined on the graph, you project it into this basis function. Then you filter it in the spectral domain on the graph in order to obtain the coupled and decoupled portion, and you can quantify coupling, decoupling at the nodal level with structural decoupling index, or at the edge level, obtaining two connectomes coupled. Now the findings. So what did we find out in these past few years by looking at this at these metrics? So first first attempt was on resting state at the population level. So we have a certain number of subjects. Here it was HCP data, 56 subjects. What happens if we average everything and we look at if there is an average picture, let's say. So are there really some nodes that are more consistently coupled and some nodes that are more consistently coupled? Well, it looks so, and what we obtain, so this is the SDI, but SDI average across the population. You see that the pattern that we are saying is really not random, right? So the nodes that you see as blue, these are more coupled to the structure, and they end up to be really the primary sensory network. So the visual network, the temporal, so the auditory network, and the somatomotor network. So these are more basic networks, let's say, the one that activate when you have a visual stimulus and so. Otherwise, regions that are more decoupled, so the red ones, they belong more to a higher level cognitive function like memory, emotion, and so on. So first, we link this pattern with previous analysis that showed that linked basically each region of the brain to a specific function, and we showed that these patterns have a behavioral relevance. So we go indeed from lower level function, more coupled, to higher level functions that are more decoupled. Again, we have this opposition between primary sensory and higher level cognitive regions. But also what was interesting to see, so the first one here is called like a behavioral relevance meta-analysis. So we find this match with the behavior, but also we find a match with a pattern of temporal hierarchy that was found before based on completely different techniques. This is a genetic study that basically looks at what are the proteins used for neural transmission. And there are some, so the blue ones are proteins that are more used for longer sustained activation. And so this is this is faster, and this is longer activation. Okay, so here we can speculate a bit, usually for interpreting results. And because, fully data driven, there is this perfect match between the regions that are more coupled for us that would also use protein that act faster. We can speculate that, okay, if the brain needs to do something very fast, then it will use the direct path. It's like taking the highway, right? I will rely on my function. And this is typical of the primary sensory network. You see something, you need to react, and so on. Otherwise, if you're doing more complex tasks, more higher level cognitive, then on one hand, we are saying, your brain can afford also to, it doesn't have this very strict temporal constraint. And what we end up seeing is that it can afford using more indirected paths. That is basically what we are encoding with the behavioral and decoupled function. So this was the first very interesting thing to see. Then in further studies, we looked at what happened when we are doing a task, for example, so not resting state here. Again, on HCP data, we analyzed, there are 7 different tasks that the subjects are doing on 2 consecutive days with 2 acquisition, test, retest. And so here we can actually look for task decoding signature, but also fingerprinting. What is fingerprinting is basically the capability of identifying a subject only based on their functional connectome. So this has been established for a decade now in our field. So you can identify uniquely a subject with very high accuracy by looking at the functional connectivity value. Here we wanted to see if the structure function values also encode this information and how they perform in terms of both task decoding and fingerprinting. So we apply different classifications with different cross-validation settings for the two tasks of fingerprinting and task decoding. And interestingly, what we see is also we do it separately for 5 different metrics. So the FC is just for reference, right? Because this is well known. So we use functional connectivity nodal strength as a measure at the node level to be able to compare it to the nodal SDI, okay, structural decoupling index, then the conventional functional connectome, and the coupled and decoupled. Okay. What do we find? In terms of task decoding accuracy, okay, it is very high at the edge level. Okay, so even if the pattern look actually kind of the same when you average them, there is still a difference that allows you to actually recognize pretty well the task. And we can reach a higher performance with nodal SDI with respect to the respective nodal measure function connectome. But also more interestingly, basically when we look at fingerprinting, again, we kind of expect that is very high because we already know that based on functional connectome is really super high. The interesting information here to add is that the best accuracy is reached for the decoupled portion of functional connectivity, which was the one that we said, okay, doesn't make sense or not. And in order to further investigate if this was a meaningful piece of information, we correlated it with a bunch of behavioral, so the fluid intelligence scores that these subjects also have. And we can see that the decoupled FC indeed has the highest brain cognition correlation, 0.6 with respect to the others. So basically the take-home message from these results is, okay, what you encode in the coupled functional connectome or coupled measures even at the nodal level is more a signature of, yeah, maybe task decoding, what you're doing, like more of an average of the modulation, but the very unique strategy that is basically used by individuals is encoded in this other decoupled portion of the connectome. Then in the more recently, what we try to do is actually implement multimodal fingerprinting in order to look at other techniques that look at functional signals and functional activation. In this case, you have electrophysiological techniques like the EEG and MEG. Okay, and also in this work, we compared EEG and FNIRS. They all give different metrics of functional activation in your brain based on different principles. In this case, for this technique, you have a lower, unfortunately, spatial resolution. So you have less nodes because they're based on electrodes. But you have the capability for EEG and MEG to look at different temporal frequencies. So basically what is interesting here is to look at emerged analysis of different spatial frequencies, so frequencies in space and frequencies in time. So delta, theta, alpha, beta, and gamma, so this is the conventional bands that are used in order to analyze this kind of data. And we can actually look at compute. So what was done here was computing the structural decoupling index based on this functional data and see if it changes. So if the signal is more coupled or decoupled in a particular temporal frequency, actually. So the general take home could be that there is a common pattern of structure function coupling that goes across temporal frequencies, but there are also frequency dependent deviations from the mean. And this is what we are exploring in this work that we are writing now, where we also look specifically at the differences basically the deviations from the mean of the SDI. Because you see, if you look at the mean pattern across temporal frequency, it looks very similar, but maybe this mean average pattern covers what are the fluctuations. And when you remove it, and you look at the different number of frequencies, you see that some frequencies are very different, and this makes total sense. For example, alpha and gamma that happen basically that encode or that say are responsible for different functional mechanisms. They have quite opposite coupling patterns. Also, we investigated how structure function coupling changes across different functional states. In particular, just to give you I will not go into much details, but we can see that basically our structure function coupling in the brain changes with respect to the level of drowsiness. So we can do that by based on resting state by analyzing only half of the sequence. So here it was the first 7 minutes and the last 7 minutes. So it's well known that the first 7 minutes you're more alert and then you're more drowsy. And so we see different patterns of structure function coupling in these two. I mean, there are significant differences between these two. And also we can look at sleep. So we have a recent sleep study where we are recording EEG fMRI in the scanner. So EEG in the fMRI scanner. EEG in order to score the sleep. So subjects are sleeping, taking naps in the scanner. And then we can look at the fMRI time courses, but we also see what's the sleep of stage the subject were in. So either N1, N2, N3, these are non-REM sleeps, or REM, okay, which is the last one. The very unique thing about this data set is that we have REM that is very hard to acquire in the fMRI scanner, and we can see for the first time, or not for sure, the structure function coupling for the first time, what happens during this phase that is very different from the rest. And what we could see is that actually we can classify based on metrics of functional connectivity, but also structure function coupling. We can successfully classify sleep stages, and in particular, our latest result indicates that the decoupling portion of the signal is also the one that allows you for a better classification of REM with respect to the coupled one. And this goes a bit with the hypothesis that we were saying. So encoding a bit the variations and even like on these more complex stages of sleep. Okay, so where did this finding leads us? Where do we stand? So we have different strands about this that we can outline about this method. One is data-driven. As we said, there are other methods of combining structure function that one can use, but that rely on models and big computational models. Here there is no underlying model. We can explain successfully how different structural vectors give rise to different functional signals, or a function that is dynamic in time. And we can find metrics that are proven to be a signature of our brain in during resting state, during tasks, that could identify individual and that we will see also are useful in our clinical application. Limitations. So for now, there is not much about dynamics, temporal dynamics. So how does my structure function coupling changes over time, you can say. So we kind of approach this analysis, but it's still very preliminary and not clear in terms of findings. You can also do a combined space-time analysis, but that becomes more complex. We explored, as we said, EEG and MEG and multimodality, but also this could be explored further, and in particular, some issues about the spatial resolution to be solved. And also, so far, I mean, let's say it's a bit still an open question to understand if there is a clinical application that is really good to target with this approach. So to answer this last question, I wanted to just give you a quick overview of the clinical applications that we looked in the very few recent years. One that is interesting is this 22q11.2 deletion syndrome. So these are subjects that are born with this is a genetic condition, and they are diagnosed quite early because of other malformations like cardiac malformations. And so it offers us a pool of subjects that are very good to study because also subjects have increased probability to develop schizophrenia at a later stage in life, normally around 20 years old, or I mean when they reach adultness. And so here we have this data set that is longitudinal, so different visits for the patients from 7 to 34-year-old, so it's quite good. And we can correlate with behavior and look if there are structure function patterns that reflect actually the change in behavior of the subject. In this case, we did this variant analysis to correlate with age. So to see if there are patterns of structure function coupling that change over time and correlate with the age. So here you see three main components, so three main patterns. We're not going into the details, but what is very interesting to see is that these components actually encode variations that are really relevant from the clinical point of view, because you see how much this pattern occurs in each subject here, and sorry, it's a bit small. Green are healthy controls, and the purple are patients. And you can see the first one appears a bit as a pattern that is really pathological, because it increases in controls and then it decreases in patients, so it fails to occur in patient. And the switch, you have it around the age of 20, which is when we know that basically schizophrenia occurs. And the second one, instead, it's a sort of pattern with a negative offset from the start. And the third one, instead, you see it's all zero for controls. And we can say that it's a sort of compensation pattern, because it really peaks in patients, but then goes down again around the same age. So this showed to be quite useful information that you can retrieve this from. A second application is to temporal lobe epilepsy, where we can actually project a different functional signal based on EEG, on the structural connectome. And in particular here, what was projected is the interictal epileptic discharges that are the pathological signature of epilepsy patients in their neural activity. And then if you look at the energy of the coupling and decoupling during these discharges, basically, you can see these quantities and quantify it as or see them as a measure of brain segregation or brain integration, because the low frequency are more patterns that are more integrated. We said these are large scale brain patterns, and you see that in fact these kind of peak after the IED, and the high frequency instead they would encode integration, and you have a lower peak. So basically the message here, what we found out is that during these epileptic discharges, during the seizure that you would expect maybe the brain to collapse or at the level of communication, actually we show that it's integrated, it has a peak of integration, where instead of working in segregated networks that might be useful, it's a sort of reset in last, this is really like few words on the last application that we are exploring right now. It's about stroke. These are patients with the upper limb motor stroke, and it is interesting to look at this subject, because stroke, basically you have an imparted area in the brain. It's really localized. You have a lesion. In the case of this patient, the lesion caused an impairment in the upper limb, so arm, for example. But it's still considered as a network disorder. So you have consequences of alterations in your brain networks across the whole brain. And also this infarcted area, it's like an alteration at the structural level, but it will cause alterations in function and also functional reconfiguration. So it's interesting to look at it as a network disorder, and also to assess what are these structural and functional reorganization. So do we gain something if you look at it in this combined way, but instead of looking at structure and function separately? What we are seeing here also, it's a data set where you have different time points. So you have an acute phase, T1, 1 week after the stroke, and then you have some subacute phase after 3 months. So you can look at the reconfiguration. When we can see, well, when we look at the acute phase, you have indeed a lower coupling in some regions of the brain that also confirm some previous reports in literature, and that is specific of the lesion side. So this in decreased coupling, you have it in subcortical region that are ipsi lesional, so the same side of the lesion, or in somatomotor networks, the networks that are involved contralateral. So this perfectly makes sense with their clinical behaviors, let's say. But then when you correlate it with the behavior, it's interesting because you can still find patterns of structure function coupling that are differently correlated to different behavioral outcomes, also that are non-motor. Okay, so attention, like it impairs also different areas and not only the one that is impaired motor area, let's say. But what is also interesting to do is actually trying to predict from the brain data, so structure function coupling at time point 1, what happens at time point 2, so at the subacute level in terms of behavior. And we have a significant pattern of correlation. So basically in these regions, the level of structure function coupling predicts the behavior at the time point. So showing that this could also supply with useful information for that. Okay, so this brings me to the end. And yeah, so I hope that basically what you understood from today's take home is that GSP structure function coupling method can be useful in order to analyze structure function coupling in the brain. We saw how this looks like in resting state. We saw that we are able to do fingerprinting and task decoding. We preliminarily approach what happens dynamically and when you look at different states of consciousness. And also we are exploring different clinical applications in order to see what is more useful for them. Thanks a lot for your attention. And that's it. If you have questions.
Thank you, Giulia. Thank you for the amazing talk. Actually, I have some questions, but I can ask it later. So, any questions?
I have several, but first off, thank you for the talk. This was really amazing, really great work. I'm interested in like extending some of your fingerprinting results because it suggests to me that this might be useful for looking at individual differences in the brain, given that you're reliably able to fingerprint more than like normal FC. So I'm curious if you have any like intuitions or if you have any results that speak to like individual differences or links to like behavior. I'm especially interested in mental health as well.
Into what?
Mental health.
Yeah. So I mean, the result that we have on fingerprinting really suggests that you do have specific unique information in the structural function coupling. We only analyzed as controls on that, so I think it would be very interesting to look at the clinical application on that. Yeah, basically in a few more details, what I can say is that when you see here, I mean, you see the patterns. You can look more in detail in the brain region patterns of the brain that basically correlate more with behavior. And I would say, I mean, we only explored fluid intelligence, but we'll explore other behaviors. And you see that regions that are here higher in decoupling correlate more with the positive values here, for example, sustained attention, verbal episodic memory, and you have frontoparietal here, so regions that seem to make sense, also with previous results of film, right, based on frontoparietal network being more able to predict individual differences. So I think it's a field where it's a lot still to explore, yes, actually.
I have more questions.
You can also talk later.
Yeah,
with you. Yeah.
Thank you for the talk. My question was regarding the multimodal approaches, just small query. So when you have the multimodal results, was the structural data also taken from diffusion?
Oh yeah.
Okay.
Yeah,
so
yeah.
it was.
I mean, actually, it's always multimodal because you have structure and function. But it was more to stress the concept that we also want to go beyond the functional hold and particular neurovascular coupling and see what happens if we can find this electrophysiological signature in EEG, MEG. So in this sense, exploring different modalities, but it's always projected on the structure, yes.
And the structure is from the same diffusion.
From a diffusion, yes. Yeah.
Thank you for the talk. It was very nice. I don't work with Preti, so like maybe my question gonna be a bit basic, but so I actually wanted to understand a little bit, like just I think I lost the connection at the end of the kind of the method to define it. So when you do the so you have your all your eigenvectors from the Laplacian and you classify them from low frequency, high frequency. Then these are related to the function to the graph signals, no? That maximize the smoothness, like each eigenvector.
So yes.
Yes.
So the eigenvectors maximize the smoothness
Yes.
if you take the low frequency
Yeah,
ones,
yeah.
and then you project a signal which you don't know.
Okay.
You don't have any assumption on that. If it is smooth on the graph, it is not smooth on the graph, and basically you decompose it on the eigenvectors.
Okay. Okay. And then when you go to, so then you have your decomposition with the eigenvectors, but then how do you connect? Because at the end in the findings, sorry, the 24, for example, is like 24. So you have each region which is
Yeah.
decoupled or coupled.
Yeah. So this
How
is
do you
exactly
connect?
this.
Okay.
This one. Do you remember we said
Okay.
we're going to quantify at the nodal level if that node, the energy, basically in high frequency versus low frequency across time. So this collapses to a nodal value, the SDI.
Okay.
And this is what is plotted
Okay.
here.
Okay. Thank you. I think I lost it.
Sure, no problem.
thank you.
Okay, good to clarify.
Yeah, no, thank you. It was nice. Thank you.
I got many. So first one, how do you think about putting in autocorrelation and the temporal parts? Because I can see two entry points. One from the time signal directly, where you just assume some sort of autocorrelation in the signals in the decomposition. The other one will be changing the structure connectome, as in not using the connectome directly, but using say powers of the connectome. Yes, that you're having, what is affected steps 2, 3, 4, and so on. I wonder whether you had anything there.
Yeah, no.
Then the second one, so I think a lot of times, sorry, is that everything here is based on spatial decomposition, right? Which is the standard thing. But there is a lot of work, actually not that much in the neurospace, but about a little bit about low dimensional factors being dominant in terms of prediction of the signals. So one could actually do the fully dynamical decomposition without having structure. I was wondering whether you thought about combining the two, as in looking at how much of the, let's say, spatial frequencies talk about the actual dynamical eigenvalues there, eigenspaces.
So, okay, let's start from first.
Yeah.
You said, looking at the autocorrelation of temporal
Yes.
you mean?
Yeah.
Okay.
Because right now everything is like.
Right now every time point is considered independent.
Yeah.
This is obviously a pity, right? Because we know, but it's a bit the generic idea of diversity in fMRI.
Yeah.
And it's really rare to have methods that incorporate this information, like autoregressive models and things, but they're not so spread, let's say. So it's in this sense equivalent to the functional connectivity.
Exactly.
We tried actually to do something on that direction in order, for example, to try to predict the next time point. So using the Laplacian, the spatial diffusion, but also as a temporal diffusion, let's say, which is a bit tricky, methodologically not easy. So we didn't do, but we can work on that. I mean, I think there would be potential, but we didn't yet concretize anything in that direction. Also powers of the structure, I mean, going from like different powers. I mean, that as well. We tried with the second. But, it's not that it didn't work, it's just that becomes very complex in terms of interpretation, which is already, and then it becomes more difficult to understand where you're going, to have a clear hypothesis. But again, there's a lot of there's nothing second was
you have the full spatial decomposition and use
on the structure.
Yeah.
Yes.
And you could do like SPD of the signals directly, and then you have factors on one dimension, spatial nodes on the others, and you could look at which talk to each other.
So you mean you can find harmonics from functional?
Yeah, basically yes, right?
Functional harmonics, and then compare them.
Yes.
And then compare the.
And I was wondering whether you looked at that and whether there is the trivial mapping, like slow frequency, spatial frequency, like small spatial frequencies go with like small signals or not.
Okay. So you mean temporal frequencies and see if they match. Yeah, we didn't do that. Also, there is the catch that in fMRI normally you focus on low frequencies. So you have this 0.01, 0.15 bandpass filter. and you don't have that many, but this could be
Sorry,
from the idea of
Giulia.
them. Yeah. But yeah, no, we didn't explore that. But so everything you see here is a bit based on the low temporal frequencies, let's say, that you know, and yeah, that drive a bit resting state.
Yes.
I was sort of thinking when you were talking about the lower spatial frequencies about like work through Rick Betzel's lab looking at edge time series, and they've shown that like in the moments where you see sort of the entire brain activating together, those are the moments that like drive the correlation coefficient that we use in functional connectivity. And so I'm curious, sort of to link that back to your work, if you've looked at the spectral waves that you get in combining the functional and structural data, like do you see something interesting in the low spatial frequency waves?
Yes, but you mean specifically in these time points? Or we didn't try, but actually it would be quite cool in order to match the two.
Yeah.
Because this goes more to the direction of selecting some peaks of interesting information, right? So some time points and not all of them. So you could, for example, do the analysis on those time points and see how it differentiates, because this is per time point, right? So you can definitely. I don't know if you meant, like, if you look at the spectrum of spatial frequencies, like low versus high. I mean, I think I have an extra slide because I didn't show it, but yeah. So basically, if you project your functional data at all the time points and then average on the harmonics, on the structural harmonics, still spatial frequencies here, you can see this sort of power law behavior. So, I mean, this is also quite expected, but it was nice to see the first time. So as we were saying, what is mostly driving the signal is this low spatial frequencies, these large scale brain networks. They are more present in terms of energy. And if we do, because when we split the spectrum, we actually do the median split criterion. So we look at the energy that is the same, that it splits the spectrum in two halves. The first 30 equals the last 330. So you see, like, high spatial frequency, they are used much less, each of them. Because, I mean, we know how the average behavior is. But yeah, was it a bit what you were...
Yeah.
We got another one, last one, because we have been talking to people like Keith Hanger, Julia Shrew, and so on, and they're like the resurgence of the whole verificality idea done great. And the results you're showing in sleep, they seem to sort of align. I wonder whether they align. So what they're seeing is that the longer you're awake, the further away from sort of behavior in terms of the properties that they find with the signals are. And in your case, you were seeing that during sleep, you were going back towards low spatial modes, right?
Low frequency spatial modes, especially in the drowsiness, not in the sleep. Because in the sleep, we didn't really investigate that yet in terms of modes, just in terms of classification. But in the drowsiness, it seems you have this behavior, right? Yes, so it's sort of default state. So if you're drowsy, and this is also shown by other, like Andrea Lupi works on different states of consciousness. When you compare, for example, anesthesia to psychedelics, so like when you go from one to the other, the brain goes towards more coupled states. So the coupled, the low spatial frequency dominate in this more drowsy or lower level of consciousness with respect to the
so that leads to the guest for those things. Okay.
Okay.
No, it's fine.
So yeah, I mean, here what you see is patterns of variation from the mean SDI. So it's not like the SDI like that, but it's like what it would mean is here you have all variations towards more coupling. So these regions, they are more coupled than normal, whatever is their average, if it's more. But yeah. Sorry.
No, it's fine. We should talk about that with people.
Any other questions? Any questions online? No? Okay. I have a question. Okay. Why did you decompose the brain signal into low frequency, high frequency? and the high frequency part is related to the decoupled portion. So how do you tell that all this information are useful but not noise?
Yeah, that's a very good question, actually. That would be the first thought, right?
Yeah.
This is just a noise.
Yeah.
If I'm right. And you're partly correct. I mean, not 100% otherwise. But that we really wanted to make sure that this component was not noise, that this was one of the first concern, because whatever is noise, okay, the data are obviously denoised, preprocessed, so you try to account for that in the preprocessing, so to remove all the noise that you have. But whatever is left will certainly or very high likely go here. You're right, because I mean the noise you don't expect it to be coupled to the structure. Okay? For example, the typical noise that you have also in fMRI artifacts and so on. So I mean, yes, it's a concern, but then I think what makes it what makes us safe from that is then showing that indeed there is a meaning in these patterns that we find. So related to behavior, able to decode individuals and so on.
So you have actual evidence to prove that they are not the noise.
Yeah, also the fact that when you look at the ratio between this energy, it's really like a behavioral gradient, if I'm allowed to say this word. So it's really like let's say a pattern that goes from lower level to higher level behavior. So the fact that there is this link, it's reassuring. But yeah, I mean, obviously the data should process for noise. Otherwise, you would have it not in the decoupled.
Thank you. And I have another question. So the Preti noise is more like a mental dynamic, right? So how much of the main activity subspace will you think sits in the low frequency parts and in the high frequency parts?
How much of the activity?
The main activity
The main activity?
about the brain signal, or we say the dynamic.
Yes. Do you mean if it does it relate to global signal? So whatever signal you have in the fMRI part of the global spatial signal, or it's not that just?
It's kind of like the
So okay. Maybe I answer it and then you tell me if it's what you were looking for. So here, when you look at let's take a decomposition here, yeah. So okay, when you decompose your signal into there, it's okay. You mean you can have fluctuations here in time, but you also have an average, like the global signal spatial that sometimes regress out or not. So it was not regressed out in here, so it is in there. But if we do it with global signal regression, basically what you have, I mean, it is captured by this global component here.
okay.
Because you have a sort of descent in your decomposition, this kind of trivial constant eigenvector that captures a bit the mean spatial variations in here, and then what is left there is what dynamically changes apart from this. But I don't know if you meant, did you mean the first time? Global?
Okay, I think I got my answer.
Okay.
Thank you. Thank you. Any other questions? No?
Yes, but we can chat at lunch.
Yeah.
Yeah.
You guys are great. So you're so much interested.
Okay, so thank you, Giulia. That's online for attending our seminar. Thank you. And thank you for the other paper person. Thank you.
Thank you.